Hi everybody. Today I'm going to show you a 2-point test cross problem. This is taken from the textbook, and we're going to do problem 7.8. 7.7 is answered in the back of the book, so I would suggest after you look over this video and see how I do 7.8, you want to go back and try 7.7 and see if you can get it to work out. So in this problem, they're telling us that genes A and B are 20 centimorgans apart, and that we have a particular genotype. Now, remember that we're not doing this one. We're doing what they say down here in problem 7.8. So first they say diagram the cross. Now remember, with our linkage sort of a writing, when we say, for instance, in this problem, A plus B slash A plus B, what we're saying is that we know on which chromosome in the organism these alleles lie. And so we have one chromosome and then the homologous chromosome. And we're crossing that by an individual that is AB plus slash AB plus. Now, in traditional Mendelian genetics, we would say the first organism is homozygous dominant for A and homozygous recessive for B, and vice versa for the other one. The problem with that here is that the genes are linked, and so we need to consider where physically on the chromosome each one of these are. So these are the parents here for problem 7.8. Part A says diagram the cross and show the gametes produced by each parent and the genotype of the F1. So the parental gametes in this case, this parent can only give us the A plus allele and the B recessive allele. It doesn't matter if we have crossing over or not in meiosis, they only have that information to pass on. And similarly, the second parent only has that information to pass on. When those combine to give us the F1 organism, we will have A plus B on one chromosome and A, B plus on the other chromosome. I'm going to jump forward to part D. Is this an example of coupling or repulsion linkage phase? This is repulsion, because the wild type, or the plus allele for each gene, is on a different piece of DNA. If I had A plus B plus on one piece of DNA, I'd call that the coupling heterozygote. But this is the repulsion heterozygote. So what gametes can the F1 produce? So here's my F1 gametes. I can produce 4 of them, just like a normal case, when I would say A plus B or AB plus. Those are what we'd call the parental type gametes, because they're exactly the same gene organization as the parents. The other 2 that I'm going to get would occur if I had a crossing over event that happened to occur in between there, when I would get A plus B plus for one and the converse AB for the other. So I have my parental type here, and I have my recombinants here. They say with what proportions? Well, if these 2 genes were unlinked, like in a normal Mendelian cross, we would say 1/4 chance of each, 25% each. In this case, now when we look at proportions, this is where we have to look at the distance that's been given to us. Remember that a centimorgan distance, in this case 20 centimorgans, means that the recombination frequency is that number as a percentage. That means that our recombination frequency here is 20%, which must mean that our frequency of parental chromosomes is 80%. And since the two different parental chromosomes come from the same meiosis event, they have to be equal. And the same thing for the recombination event. I expressed those in decimals because we're going to end up multiplying them together later, and it's helpful to have decimals. Okay. So when they ask us in part C, if the F1 is crossed to a test cross individual, something totally recessive, what offspring would be expected and in what proportions? Well, now we can go over here and we'll make ourselves a simple Punnett square where we say I have the 4 possibilities from the F1, just like we would otherwise for a normal Mendelian cross. From the test cross partner, of course, I can only get AB recessive. The difference here is in the proportions. Each column is not the same. I've got a 40% chance of having a gamete corresponding to one of these first 2 columns, and only a 10% chance corresponding to the second 2 columns. The test cross individual, though, 100% chance. There's only recessive genes there. And so I'm going to go ahead and maintain the linkage as I write these. I'm simplifying it to 1 line just to not crowd this too much. And I'm going to do the same thing I did for Punnett squares before, where I bring the alleles from the top and the alleles from the side together. And I get my 4 different genotypes for my offspring. If this were normal, unlinked, or independently assorting genetics, I'd say 25% each of these 4 things. In this case, what I'll do is I'll multiply my percentages together to get the percentage of each category. So 0.4 times 1, 40% of the offspring should look like this, and 40% of the offspring should look like that. 10% of the offspring should look like each one of these. Now, in some problems, we might know what gene A and gene B do, so we might have descriptors. In this case, we just know it's A plus, B plus, or whatever it might end up to be. So that's a test cross. Test crosses are important for us because they allow us to determine distance by counting offspring. We'll see some problems like that in the book. This one's going the other way around. We know how far apart the genes are, and we're trying to calculate percentages of offspring. Now, how does it change if we're dealing with an intercross, part E? Well, I'll go ahead and erase some of this so we have some room to work. But basically, an intercross is still going to be a Punnett square. It's still going to be a combination of the alleles across the top and the alleles from the bottom. But what we'll do, I hope I have enough room here. We'll do in the case of an intercross is we'll still put our alleles that we can get across the top. And it doesn't matter what order you write them in, but I'm trying to be consistent here. And we'll write our alleles down the side. This would be the other one of our intercross. It's again another F1 organism. The Punnett square is exactly like what we've done before. And if we do this, the fun part is we know what the ratio is going to be. Back in chapter 3, we looked at Mendelian genetics, and we know that when we do an intercross for a dihybrid, 2 heterozygotes, we know that we're going to get 9 of our boxes that are one type, 3, let's do it this way, 3 that show each of the intermediate phenotypes, and 1 that's totally recessive. Now this is telling us how many boxes out of 16 we're going to have. And that's still true when we talk about phenotypes. The problem is, in a linked case, each one of our boxes aren't worth the same part of our percentage. So we can look for 9 to 3 to 3 to 1 in the boxes, but not in our total proportions. Because remember, being in the first column or the second column gave us a 40% chance each, but the recombination events were 10%. Same thing is true down the side. 40%, 40%, 10%, 10%, because of the linkage. So now when I go through and I fill in my phenotypes, or even genotypes, I'm just going to run out of space here, I can say, this one, for instance, is one of my A plus, B minus kind of phenotypes. It would have fit into that category. That's one of those 3 boxes. If I look for my other ones that are A plus, B minus, where am I going to find those? I'm going to find them here, where I go A plus B from the top and AB from the side. That also would be an A plus B minus phenotype. And I'll find it over here, where I go A plus B from the side and AB from the top. Those are my 3 boxes that would have fit into this A plus B minus phenotype category. To get the percentage for each one, I'm going to go ahead and multiply the percentage from the top times the percentage for the side. So 0.4 times 0.4 is 0.16. 0.4 times 0.1 is 0.04, and 0.4 times 0.01 is 0.04. So this phenotypic category that I've sort of focused in on now has 0.16, 0.04, 0.04. It's 24% of the total. It's no longer the 3/16 I would have predicted from Mendelian genetics. It's a little bit higher percentage than that because of the linkage. If I go through and I do the same kind of an analysis for this, I maybe look for my 1 AB recessive. We'll find out where that guy is. So he's going to be here, where I say AB recessive from the top, AB recessive from the side. There's still only 1 box that contains that phenotype like I predicted, but now its probability is not 1/16 of the total, it'd be about 6%. It's actually 1% of the total. And I got that by doing the 0.1%, or 0.1 fraction from the left times the 0.1 fraction from above. If I look for my A minus B pluses, I can find those in here. AB plus from the top and AB plus from the side. I can find it here. And I can find it over here. And again, I'll do the percentages the same way. It's still 3 boxes, which is what I predicted, but now 0.14 times 0.14 for the first box, and 0.4 times 0.1 for each of the other boxes. So I see that that one's a little bit more highly represented than I would have expected. And then lastly, the other 9 boxes that I'm missing. So 1, 2, 3, 4, 5, 6, 7, 8, 9. Those are all going to be doubly dominant phenotypes. I can go through and calculate the percentage for each one based on taking the percent from the top times the percent from the side for each box. And I can add those all together. Or I can simply say it's 1 minus everything else. So 1 minus 0.24 for the blue category, minus 0.24 for the green category, minus 0.01. And if I've done that correctly, I think I'm going to get 51% of the total. So in this way, we see that I do still get the 9 to 3 to 3 to 1 graphical representation, but since each box is weighted differently, I get a different fraction. So in part E, when they say if the F1s are intercrossed, what offspring would be expected and in what proportions? Well, we would expect more than normal or more than Mendelian ratios for each of the parental types. Remember, these are ones that contain the same sort of a phenotypic arrangement or genotypic arrangement as our parents in this cross. And we expect a lower than Mendelian for the ones that were recombinant. This difference or this alteration from Mendelian ratios gets more dramatic the closer the genes are together. That's what we call map distance or frequency of recombination. But it's always the same idea. So the twist here is that when you build your Punnett square or you do your forked line method, you're not going to be using the traditional 1/2 or 1/4 or that sort of a thing. You'll be using the distances given to you in the problem.